be organized with a view to enhancing the academic and research programs of higher education in our country. We know that there are over 3 million Sri Lankans living abroad and fair proportion of them holding senior positions in academia and R&D institutions and some also in the industry. They are very keen to contribute, share their knowledge and experience with the motherland, but there has been no credible and pragmatic mechanism for them to contribute. Because of this reason, the National Science Foundation constructed a digital platform to harness this hitherto almost untapped and untouched intellectual asset and resource for the national development, and also to offer opportunities from our end so that there would be mutually rewarding and reinforcing partnership. Today we are living in a globalized environment where quality and relevance, when we talk about quality relevance, we can talk about Sri Lankan standard or SAARC standard or Asian. It has to be global, international standard. And we would like our scientists to occupy on the frontiers of knowledge and take a share in moving them forward. Confucius said, by birth, people are same. Through nurture, they become distinct. So here, the purpose of this, that means whether you are born in USA, Ethiopia, Canada, or Sri Lanka, inherently at birth you are same. But your blossoming depends on how you are nurtured and fostered. That is why NSF, trying to create that atmosphere, decided to really harness these intellectual assets for the benefit of our motherland.
Thank you, Professor Senaratne, for having me in this forum to talk to you all. And I'm also, like Dilant, a proud product of Peradeniya Science Faculty, thanks to my teachers and the mentors. Now, we all heard how we need to move and change our research culture in Sri Lanka in the previous talks. Now I'm going to talk about what I can offer, what I'm doing here, and what I can offer to the Sri Lankan student and scientist while I am here. I was, before joining to Texas, I was in University of Mississippi for 10 years, where I developed a plant science, especially on medicinal plant program. And then they invited me to join here in Texas to develop the similar program. I moved to Texas in early 2013. So a good thing in Texas is is 5 times bigger than Sri Lanka, and we have subtropical climate. So I feel more like Sri Lanka, and we can use a lot of the plants to grow here like in Sri Lanka. So I enjoy what I'm doing, and so I will move into the presentation, and I'm going to discuss. This is my short outline. I'm not going to talk too much. My research capacity in the Prairie View, and Prairie View is one of the member universities in the Texas A&M system, where we have 14 universities. We work together in research and academics. So I teach in various universities while doing collaborative research with different universities. So I talk about the capacity in my the College of Agriculture, and the research highlights I have currently, and then the availability of opportunities in my department. Right. When we look at the capacity in the plant and environmental sciences here, we have 780 research farm, the acres of 780 research farms, so that allow us to do a lot of various agriculture research and basic science research in the field. And we have a huge collection of greenhouses. They are climatic controlled and grow chambers. And the new addition is the Pheno AI expert system, where you can do the phenotyping and use artificial intelligence for plant phenotyping to understand their growth throughout the growth cycles and to characterize them. And then I have a core lab complex, which includes genomics, analytical, and microbiological facilities. And apart from that, we are maintaining a huge germplasm collection of medicinal plants from all over the world. And also, we have underutilized crop species collection, which we are trying to further develop the various crops. And recently, we added the cannabis also into our research line. Right. So the plant and environmental science department has these the key research areas, and I'm actually the trained in plant systematics, and I started my career with Professor Dasanaik and Dr. Cyril Vijayasundara in Royal Botanic Gardens, Peradeniya, writing the flora of Ceylon, and then I moved into my doctorate on Annonaceae systematics. But when you look at the pure systematics, there is no much use in it. So I had to expand my horizons. So I switched on to medicinal plants and characterizing and the applied part of the science in it. So I mainly focus on systematics. I still work on systematics on various parts of the world in floristic revisions, and the mainly focus on the research is mainly focused here in the university is towards the medicinal plants and underutilized the crops because in the US, as you know, the healthcare cost is very high, and the people think the natural way of maintaining health by using plant-based healthy product is better for your health. But it is not true all the time because most of these plants are growing in the one part of the world, but the products they develop here in the US. So the raw material comes from somewhere else, and we have no idea about the safety, quality, and efficacy of these products. So by having the living medicinal plant collection, we will be able to characterize the medicinal plants using genomic, chemical, and botanical data, and then compare the quality and safety of the products using those markers. So one of my research labs are mainly focusing on working with the US FDA to evaluate the quality of the plant-based health the product. And then we have a plant biochemistry, molecular biology group, polymer chemistry, phytochemistry, plant breeding, and we have specialty crop and urban agriculture, plant physiology, mainly on the crop branching patterns on the major crops, and biotechnology and agronomy, bioinformatics, and then the environmental team has water and natural resource management, and they also focus on global climatic change, remote sensing. So let's look at some of the key work we are doing here. Now, the Living Germplasm Collection and Seed Bank has over 2,000 accessions at the moment, and we are still exploring the plants through the collaborations with other countries. And currently, we are focusing on two plant species, and this actually has already completed the working on this plant, the phyllodialysis. We are looking at the natural sweetness because obesity is one of the big problem in the US, and the consumption of high calorie food and the sugar is a major the problem. So USDA funded us to find out the natural sweetness and try to introduce them to the industry in the food industry. So when I was doing fieldwork in Peru, I found this plant, and it has a natural sweetener. This is known compound Hernandezine, and but the problem in this plant was the plant itself producing the aromatic compound called camphor. You all know kapuru, like, right? So it is kind of bitter and masking the sweetener, the sweetness taste. So what we did was, this is before the CRISPR coming into the stage. So what we did was we looked at the biosynthetic pathways of the Hernandezine production and the camphor production, and then by fine tuning the micronutrients of this the plant we provide, we managed to switch off the genes of camphor production. And then we managed to get the higher Hernandezine production in this plant, and we got patent couple of months ago for this method. Now this plant, the Hernandezine, is about 1,000 times sweeter than sugar. And now we are working with the industry like Coca-Cola to include the Hernandezine into their product. And also we have introduced this plant as cash crop, novel cash crop for the Texas farmers. And the other one, we are currently working on this. This is called monkey fruit, and coming from China. It can be grown here in Texas, and we grow this picture is from our collection. And this has another sweetener called mogroside, and but it has some interfering compounds, and we are working towards suppressing those other unwanted compounds to make it more productive in the natural sweetener to introduce into the mainstream in the industry. And there are a lot of underutilized edible medicinal species. Now, these are not new to the Sri Lanka. You see the Momodica cochinchiensis, Karavila, Tuba Labu, and Dandila yam. They have never been grown in Texas. But as you know, the Houston area is multicultural, right? And people are looking for this kind of, they are missing these the vegetables. So what we did was we started growing them and introducing to the full culture. And as you know, these are also having a lot of medicinal properties. And we are working with the Texas medical institutes to find out get the clinical studies done on the effect of these the crops. And some of these crops are now in the hands of the specialty fruit growers in Texas, and especially Momodica dawesia, which we had the spine gourd or the Tumbakaravila, we only had in Indian shops as the frozen the veggie section. Now you can get the fresh Momodica dawesia and we have extension program to introduce these novel edible fruits to the American food culture, and Americans started eating them. So now you see we are getting ready for the food shortage, the challenge in the future, adding more novel crops and novel food, plant-based food into this system. So this research also heavily funded by USDA, right? That is one. And the new one is the cannabis. As you know, now the Texas got the license to grow non-narcotic cannabis that is called industrial hemp. So this doesn't give you any high, and the THC level is below 0.3 percent, right? So, but the problem is now, as you know, since the cannabis was banned in the world, the cannabis was landed in the hand of illegal growers. So the plant has been bred more and more to get the narcotic effect than the other good the factors in the plant. So when you look at the genetics, even in the industrial hemp, it is very, very reticulate and very complex. So we started developing a germplasm collection of all the cannabis strains, non-narcotic strains available in whole US, and we are now the leading cannabis germplasm collection and the leading cannabis research group in the Texas. And as you know, Texas has a lot of agricultural land, and we got a lot of interest from the farmers, industry to work with us. And I will show you some of the exciting work we are doing with the industry on this plan. Now, I know Sri Lanka has a lot of debate on cannabis legalization, and all these debates are because they don't understand really what we can get from cannabis, and they always fear about the narcotic type. Now, we are not growing anything narcotic here. We all look at the non-narcotic species and how we can use it in the industry and the agriculture, right? So we are characterizing our whole cannabis collection here using phenomic, genomic, and chemical fingerprinting, and then we will identify the best performing strains, and then we will do field trials for those strains, and then recommend for the Texas Department of Agriculture to the best strain so that they can guide the farmers how to grow the best strains for the Texas. And also the mainly the non-narcotic industrial hemp is mainly used for either fiber or the phytochemicals, mainly the cannabinoids or the seed grains, right? The fiber is used in various applications from water repellent cloth to the house, what is called is hempcrete. It has a bast fiber that can convert into the hempcrete, and now the houses are built using the hemp-based concrete. And the cannabinoids are mainly the CBD, the phytocannabinoid that has a lot of medicinal properties because it mimics our endocannabinoids in the human body. Endocannabinoids are the messengers help you to keep the body homeostasis, and when you lose the homeostasis, you know you will get a lot of various diseases, and with the age you will get the lower endocannabinoid production, and when you have external phytocannabinoids like this, you can maintain better health. Now, the FDA is doing research with us to add in these phytocannabinoids into the various food products because it improves the body function. It is going to be added into the milk like vitamin D and added into the cereals and so on. And the seed grain has a lot of omega-3 and omega-6 fatty acid and a lot of protein. And as you know now, the food culture is changing a lot. People are rejecting animal-based milk because some of the sugars in the milk is supporting the cancer growth, certain cancer growth, and now they are moving into the plant-based milk. So now you see here in the grocery stores lot of various plant-based milk products, and the hemp milk is one of the new additions. So we are working with the interdisciplinary team, food scientists, chemists, regulators, to make this industry sustainable in the Texas. And when you look at the plant growth, cannabinoids produced only in the female plant trichomes, and those are so the if the crop is growing for the purely for the cannabinoid production, so you need to get in only to the female plants grown into the field. If you get a single male plant, that can ruin the cannabinoid production, because cannabinoid is made in this plant to attract the improve the pollination. So if you have male plant and the pollen is landed in the female plant, it reduces the cannabinoid production in the plant. So we are working on the looking at the molecular mechanism of sex expression, and we recently used the CRISPR method to knock off some of the flowering genes, and we are halfway in the process of that. And when it comes to the male plants, now male plants give the best fiber. As you know, the cannabis is the wind pollinating plant species. So the plants need to grow taller to release their pollen into the air. So in the field, you can see the male plants are emerging more than the female plants. So, but after they finish the flowering, all the energy pumped into the fiber production stopped. So the fiber quality goes down. So we thought, if we stop the male plant flowering, we can increase the biomass and the fiber production. So we did the bioengineering. We used CRISPR and knocked off the flowering gene in male plants. And you see this picture I took yesterday. Actually, this is 2-week-old plant. Now these plants are about 2-3 months old plants, and you see the height. It is reaching the greenhouse, the roof. It is about 10 foot and not flowering. So we are still waiting. If it is, we still don't know. We have knocked the right gene, but it is growing very fast and giving very good biomass, right? And we have developed under this. This became very hot topic in the Texas soon after they legalized it here last year. So a lot of private companies jumped and donate money for our research to address some of their questions. We got a very, very strong private partnerships. And also when we look at certain the strains we have, we found some interesting facts. I just want to show here. Now, usually the cannabinoid is produced in the flower buds, and some plants started expressing into the younger leaves also, which has not reported before. Now we are looking at the molecular mechanism of this one also, then we can get clear understanding why it is happening in various the strains, right? So let's move into the now, this is how we have, these are the few strong companies. We develop private partnerships, Aiden Green, and they are one of the leading indoor production company. We have another project going on with them, and I will show you what we are doing in that part. And Pharmacopoeia, this group donated 18 million for us for research and student training on on industrial hemp. And we have another company dedicated to collection of data and making the data-driven decision making for all farmers, breeders, researchers, and also the government agencies who control this crop. And we have canna farms working with the product development for animal production, the cows, chicken, and the hemp. When you extract the hemp CBD, there is a big group of flavonoids and terpenes there, which we can use in various other product development. As you know, the terpenes are the compounds used in plant signaling, and we use some of these terpenes to improve the growth properties of certain crop species in greenhouse settings. And also we use the flavonoids in poultry, the cows, meals, to improve their meat quality and the egg quality and their reproductive the quality and so on, because all metazoans has this endocannabinoid system. So it works as it works in the human. So we found it improves the animal health too. We have a huge veterinary team also working on that aspect. And recently we started the MOU with the BioCity Hemp Company, the largest hemp extracting company in the Houston, Texas. They receive all the biomass from the farmers of hemp growing in Texas, and then they extract CBD, and they will sell the pure CBD for the clinicals and various other the products, and they offered us the byproducts for further product development research. So what I found was there are a lot of wax they remove in the process, and I brought them to the lab and look at the composition, and I found that there are a lot of antibacterial, antiviral, antifungal compounds in it. So we started using that wax as the protective coating for perishable fruits and vegetables. Now, currently, here, the apples, pears, bell pepper, like all these perishable fruits and vegetables are coated with paraffin wax, right? It is petroleum wax. So we got recently developed the hemp-based edible wax for this kind of application, and we are working with them to introduce that to the market. And there is a big association making the connections of the hemp farmers, researchers, and the industry. So we are working with them. Um, very, very effectively. We conduct workshops, seminars, webinars, and all sort of the technology transfer platforms to the audiences around us. And these are some of the products available in the market now. Now, we can grow, even in Texas, we can grow hemp in the summer season. It will die in the winter season. This is where the Sri Lanka can move for the industrial hemp. But just remove the narcotic part, and you need to characterize or whatever available the germplasm in the Sri Lanka, and focus on the non-narcotic ones to develop new crops. One of the medical scientists working with me in the Texas Medical Center has recently developed the heart stent and the fallopian stent using the hemp fibers. And also she uses these fibers as a scaffolding for the burn injuries to grow the cells over it, because these fibers won't give any rejection or adverse effect. It is bio polymer material, right? So there are a lot of industries, a lot of opportunities to explore, and we are still working on this part of the crop, right? And apart from that, you will see some of the green vegetables, and we in American food culture, not like ours, they only eat selected greens. So that means they are not getting enough interesting secondary metabolites, plant-based secondary metabolites through their food. So there is a big demand for now for the medicinally important greens. So we started introducing, like, the niviti, and from Egypt, this is Egyptian spinach. This is Malabar spinach we call niviti. And I think we have this species in Sri Lanka as wild, and no one is eating, but this is very popular vegetable in the Arabic culture, that is called Egyptian spinach. And we have New Zealand spinach, and the purslane, the portulaca, we have as a weed in Sri Lanka, but we rarely eat, right? But they have a lot of good phytochemicals help to maintain our health. So we introduce all these things, and we recently introduced moringa also here. Moringa grows very well in Texas now. I have 2 trees in my backyard also. And these are novel species we introduced to Texas farmers, and these are very highly priced in the market, and most of the grocery stores carry these specialty crop section demonstrating how to cook and what are the health benefits. So it receives a lot of attention in the local communities, right? So this is how, this is nothing new to the Sri Lankans, but this make a big impact in our research here, and USDA continuously funding this research. And now this is my 7th year for this type of research. And very recently we started working with the NASA because now they are moving towards space agriculture and space travels. You need to have low volume, high nutritious plant species to eat when you are in space travel and space, in settlements. So we started growing microgreens and try to improve their the secondary metabolite production by manipulating various light regimes. So we found the novel blue and red LED lights can hugely change the nutrient value in these various microgreens. As you know, the secondary metabolites means the taste, aroma, and the color, and those are produced in the plants as a response to various stress, or sometimes to attract other organisms or deter the other organisms. But they are very helpful for our health, because that is how we have evolved. The humans evolved with the plants, and how we ended up eating plants, and how we selected various plant species into our food culture. So this has become a hot topic, and very recently got a good amount of money from NASA to study the microgreens. And now the microgreens are sold in the high-end restaurants, and they are very pricey. Microgreens is not the sprout; they are little older than the sprout. So you let the two true leaves emerge from the seedling, and then you harvest them and pack them. The good thing in microgreen is you can grow them in the selling clamshell itself. So the live plant can sold in the market, and you can take it as a live plant, and when you're ready to eat, you can harvest in your kitchen and use it. So they are very highly nutrition. And when you look at the mature plant and the microgreen, the microgreen has secondary metabolites 40 times higher than in the mature plant. Now you can see how value is this kind of functional food. And also we have a separate section. We are sweet potato is not a kind of traditional crop in Texas, but one of my colleague work on sweet potato breeding, and he managed to develop purple and golden sweet potatoes suitable for Texas climate, and the paste and purple and gold we selected because that is our Prairie View University colors. It's a kind of marketing. And also we introduced edible leafy variety of sweet potato. This is also getting a lot of attention. And we also work on various biopolymers to use in agriculture. As you know, the chitin is available in the shellfish waste, and we extracted chitin from the waste, and then used it in agriculture to improve the yield and the shelf life of perishable selected the crops. So one group is working on that part of research. And we have physiologists working on the monocot crop species, and they are branching how it helps to increase the yield. So he looked at the molecular mechanisms of branching, and then manipulate the genes to improve the yield in various crops, right? And the novel thing we started recently was developing urban agriculture production, indoor agriculture production. This is what we developed with the company called Eden Green, and they developed 1 acre vertical grow facility in the university, so that our student can be trained. We do the research, and the produce goes to the local market. This is actually the leafy vegetable, mostly salad greens and the common herbs. We used to grow in this part of the world. And so what we did was we improve the taste and the spiciness, aroma, crunchiness in these greens by mimicking these plants using the byproducts of the hemp, the terpenes. We use releasing now this greenhouse is fully computerized. Every this panel has sensors, and it detects what is the right amount of fertilizer needs. This is a combined aeroponics and hydroponics going together. No chemicals used, only the aeroponics nutrient mixture, and we have small individual carbon dioxide releasing mechanism to each plant in this system. And with that carbon dioxide, we managed to include a little bit of mycene, the terpene we extracted from the hemp in this greenhouse that improve the color development and also the crispiness and the spiciness in the plant. So it is very new, and we are doing a lot of manipulations, and we are introducing various other the terpenes also, because when we damage these plants, we notice that the mycene is the major terpene released from these leafy greens. So that is the signaling. When you damage the plant release these stress chemicals to let other plants to get ready for this the danger. So we thought this is the mechanism going in the plant, and we artificially released the mycene into the system that improved the spiciness and the quality of the green. And we had taste panels, and we had chemical fingerprinting to prove what is happening inside. So these are something I was trying to introduce to Sri Lanka, because we are talking a lot of using the agrochemicals, and now trying to go into the agriculture, which I don't think is not we can do overnight. And getting Chinese organic fertilizer is very scary. I will tell you one of the very small the example. I looked at the Chinese canned vegetable products come into the US market, and I analyzed them, and I found a lot of pharmaceutical residues in the product. So I immediately informed to the FDA and found they stopped selling all these products. Then we found they are using the human waste for their agricultural products. So I'm really feared whether the Sri Lanka will be ended using the low quality the organic fertilizer coming from somewhere else, who is trying to dump their the waste. So we need to be as scientists, we need to be proactive and save the country's agriculture systematically. We can go into the organic system, but we can't change it overnight or within few years. It takes a lot of time, and the country like Sri Lanka does not have enough space. We are an island, and population is increasing, and we will not have enough agricultural lands in the future. So the best thing is to go for high throughput agriculture like this. Have one greenhouse setting with the science and artificial intelligence, robotics used in the system, and make the own fresh products to the consumers. That will help the quality product, good export market, and reducing the post-harvest losses. And we are not going to use chemicals there, right? This is chemical free, no pesticide, no fungicide, and we sell this. And this 1 acre greenhouse, we grow 5 to 10 growing cycles per year because we can manipulate the light and the environment. And from this 1 acre, we produce 286 acre worth of produce yearly. Now you can imagine how much we can generate. This Eden Green is the major producer in the US now, and it has started expanding to Australia. And also very recently, we are working with the Singapore government to establish something like this in Singapore. So these are something we can give to the country. And I was in touch with various entrepreneurs in Sri Lanka to introduce this so that we can work and we can grow our own greens in this kind of system, right? So I have another meeting in 3 minutes, so I have to leave. So these are the opportunities I can provide. I have graduate program, and feel free to contact me. And if I don't have the program fitting into your interest, then I can get you connected into the appropriate the universities and the professors. And I'm mostly looking forward to have study abroad student exchange program and postdoctoral training available. Currently, I have several Sri Lankans working in my lab, and I'm mentoring a couple of students in University of Colombo, and also can provide short-term visits for students and scientists, and open to mentoring Sri Lankan students, and also happy to collaborate with the research grants and publications. And now you got an idea what kind of science we are doing. I am a systematist, but how I use plant systematics in the applied science. So this is where we need to move. So because we are facing food shortage in the future, and so we need to feed the people globally, and the climatic change are really challenging. So there are a lot we can do together. So feel free to contact me if you have any questions. And this is my cell phone number. Feel free to contact me for any help I can give from this end. Thanks very much. Uh, I'm sorry I have to leave because I didn't expect this will go this long. And I have another private company coming to meet us for a collaborative research. So I have to leave. Thanks very much for having me in this forum. Thanks again for the NSF and the organizing team to have me, give me the opportunity to share all this information with you guys. And I'm looking forward to have very, very fruitful collaboration. I got my free education. Let me help you guys and help me, the country, back. Thanks very much.